So I think we are going to start again. So is Christina somewhere?
I'm here. Can you see me? Is my screen sharing?
Perfect. Yeah. So our next speaker is Christina Warinner from Max Planck and Harvard, and she's going to talk about the evolution and ecology of the hominid oral microbiome. Thanks.
Thank you. Well, it's a real pleasure for me to be here today, and I feel really fortunate to be speaking after Clivia and Nicholas, because they've set up my talk really, really well. I'm going to be talking today about the evolution of the microbiome, and I'm also going to be talking about a little bit about the expensive tissue hypothesis and thinking about some of the deeper questions, evolutionary questions we can address by focusing on microbes and specifically the microbiome. And so so my work in general, I'm very interested in the relationship between humans and microbes and the long history we've had with them, manipulating, living with them side by side and co-evolving with them and trying to understand this long-term relationships we have had throughout our entire evolution. Until very recently, though, our ability to study the microbiome with any kind of time depth was very limited. And really, this is about all we knew. This is about all we could find until within the last 10 to 15 years. It was really with the advent of next generation sequencing technologies that we were able to finally access the actually very rich bioarchive of DNA, ancient DNA that is available. And so the blossoming of the field of ancient metagenomics is really what's opening up a whole new view on the human past and our ability to explore our evolutionary history by looking at our closely connected microbes, our microbiome. The human microbiome itself has not been studied for that long. In fact, the word, the human microbiome, was only coined roughly 20 years ago by Joshua Lederberg, who described it as a term that signifies the ecological community of commensal, symbiotic, and pathogenic microorganisms that literally share our body space and have been all but ignored as determinants of health and disease. And the study of the microbiome and the ancient microbiome is so new that in comparison to the study of the human genome, for example, where we know so much, we know almost nothing about the evolution of our microbiome, the microbial portion of ourselves. And so one of the things I've been wanting to do, and my research team has been focusing on for the last several years, is trying to understand how our microbiome has evolved. And one of the questions we wanted to tackle is, if we take a very broad view, if we take a very broad perspective, and we focus on one of the body sites, let's say the oral microbiome, how has the oral microbiome evolved with us as anthropoid primates? How has it changed through time? How has it responded to diet? To what degree can we make inferences about changes in diet by studying the oral microbiome? To what degree does it differ between species? Does it do changes in the microbiome recapitulate our own phylogeny? Does the ecology of the microbiome track species differences? Do the individual species histories track these differences? These are all open questions to which we do not know the answers. And so we wanted to take an evolutionary approach and an ancient DNA approach by accessing the oral microbiome through time to try to address this question. We focused on anthropoid primates, so we're looking at the humans, great apes, and also howler monkeys as an outgroup. And this allows us to look at approximately the last 30 million or so years of how the oral microbiome has changed. In our study, we started with initially 124 individuals, of which later we determined 89 had sufficient preservation for analysis. And we have a distributed range of individuals. So we have 5 howler monkeys, which are really serving as our outgroup. We have 14 gorillas from 3 subspecies, 16 chimpanzees from 3 subspecies, 7 Neanderthals, and 47 humans, of which 16 are pre-agricultural, mostly to the Paleolithic and the later Stone Age. And we have 3 that we're calling pre-antibiotic, so before the 19th century, and then we have 18 that are present day. Within that data set, some of the real novel features that we have is we have among our Neanderthals, we now have successfully analyzed the oldest Neanderthal oral microbiome to date that's well preserved. That's about 100,000 years old. So our Neanderthals span about 100,000 to 40,000 years ago. And the modern humans in our study span about 30,000 years ago to the present. These are some of the examples of what we're going to be working with. How do we access the oral microbiome? Fortunately, there is a substrate called dental calculus. It's effectively calcified dental plaque that forms widely on mammalian teeth. And this calcified dental plaque serves as a bioarchive that allows us to access the microbiome through time. What we're really fortunate is that the calculus itself preserves extremely well in the archaeological record. It preserves along with the rest of the skeleton. And so this is really fortunate for us because then we can really time travel to almost any place, any time, and obtain a record of the oral microbiome. For those of you who aren't quite certain what it looks like, so this is a close-up of dental calculus on a mandible that's actually of medieval age from Europe. You can see the calculus is formed right along the gum line, which is no longer there, but you can see where the gum line was based on the distribution of the calculus. I like to use this as my advertisement for dentistry and toothbrushing. So if you never brush your teeth, you will accumulate plaque and calculus buildup over your lifetime, like this individual has. We can take one of those teeth and we can look at it in cross section, and there you can see the calculus that's sitting on the surface of the enamel in this case. And we can zoom in much more closely. And as we zoom in, you can start to see that there's some structure within it. Calculus forms incrementally, almost like tree rings or layers of an onion. The interior layer forms first by the formation of a dental plaque that then spontaneously calcifies. A new plaque layer is laid down. This process repeats. And so you're actually looking at a succession, an ordered succession of microbial life on the surface of the teeth here until this particular person, this woman, died in her late 50s. It doesn't remodel, and so it is really a kind of a bioarchive of this person's lifetime. And calculus is very interesting to me because it's the only part of your body that actually fossilizes while you're still alive. And that in part contributes to its excellent preservation in the archaeological record. We can zoom in even closer and look at individual bacterial cells in situ. So here you can see numerous bacterial cells in this particular piece of calculus. We can decalcify that matrix and the cell walls remain intact. They're actually quite robust. Here you see a gram stain from that same individual. We can then also stain, use Doppy stain or Hooke's dye to stain double-stranded DNA. We've done that here, and you can see that actually there's so much DNA present within this calculus sample that you can actually physically see it using fluorescence. You can even see in some cases the DNA is resolved to individual bacterial cells, which you see here. These are actually, it's a genomic DNA that's still contained inside of the bacterial cells. The proteins are also still in many ways of functional. They still have functional epitopes. So this is an example of using an immunoassay, an immunobinding here to show that we have functional epitopes for a particular species of bacteria. And we can use transmission electron microscopy, TEM, to look even closer. And we can see that we even have, mineralization within the cell that is probably preserving particular structures intracellularly. So what my team has done over the last several years is trying to work with this material and trying to understand how to work with it. We spent a lot of time building methods and trying to come up with ways of authenticating, analyzing, taxonomically classifying this substrate. And over that period of time, we built up an expertise and tried to how to approach this. And so this is what I want to show you today is actually some really brand new research out of my lab that I haven't presented before. On what we are finding about the evolution of the hominid oral microbiome. So as I said before, we have these different individuals that are covered here, and I want to really point out the very hard work of my student James Fellows-Yates and postdoc Irina Velsko. Much of what you're going to see in the coming slides is a lot of the hard work that they've been doing over the last couple of years. So really quickly, to do this study, it was quite complicated in terms of trying to figure out a workflow that would allow us to analyze all the different questions that we wanted to. So I've shown the workflow here, and I do not expect you to read it, but I just want to highlight the colors here. We took 4 different approaches. One was looking at preservation. The second was composition. The third was looking at phylogenetic analysis, and the fourth was looking at functional changes. So I'm going to refer to these color codes throughout. The first and most important one is preservation. Obviously, we're dealing with samples that range in age of up to 100,000 years old, and samples that come from really different contexts. So for example, our primates, although the howlers are actually come from a really well-preserved context, the gorillas and chimpanzees did suffer some taphonomic damage because they were buried for a time. They come from between the early 20th century and the mid 20th century. All of the primates that I'm going to show you in the study are wild, and this is important because captive animals have been shown to acquire human microbes in captivity, and so all of the animals within this study are wild. We have approached many different ways of trying to filter out samples that have essentially lost their original community structure and that have decomposed or degraded to the point where you can no longer analyze them from a compositional or a microbial ecology perspective. This is a technique that we've been developing. It's a cumulative frequency decay plot that actually just looks at general classes of bacteria that we know are typically associated with oral cavities in humans and mammals, and plots that, looking at the type, looking at what proportion of your community that you have and the rank that it exists matches that, or does it not match a kind of oral taxon or known oral clade. We've tested this on modern dental plaque, and we can show in blue means it passes and red means it fails. And we can show that we can develop this test that can be quite specific to identifying oral-like communities in various samples. This is when we apply it to our actual samples. And one important thing to note here is the howler monkey. The howler monkey is going to be the most evolutionarily diverged group within our set, although I said before it's very well preserved, but very evolutionarily diverged. All of our howler monkeys passed in this test, which means that our sensitivity was such that we were able to still retain those, even though they're evolutionarily distant in terms of detecting oral-like taxa. This allowed us to remove really poorly preserved samples from our sample set, in particular Neanderthals. We had quite a few that were quite decomposed, but we also had many that were very well preserved. And then importantly, our extraction control and library blanks also then failed and were removed. We have in the past also used source tracker. It's a method for using Bayesian inference to try to estimate the contributions to a given microbial community. And here we've plotted those same samples using this using source tracker, and you can see what the estimated percentages of the different sources. So either dental plaque or the gut microbiome in this case is frequently mistaken for dental plaque, and that's a kind of a technical artifact, versus various environmental signatures. We compared that to the results of our cumulative decay plots, and what I've shown here in the box is what the cumulative decay plots also indicated were poorly preserved samples, and so we removed these from our analysis. All right, and then we took another step. One thing we have noticed is that sometimes in samples, source tracker can be quite conservative and underestimate the amount of oral bacteria that are present. Oftentimes what we might have a well-preserved sample in which a certain few number of species, usually a Pseudomonas or something like that, a soil bacterium, has vastly overgrown. And this can alter the community profile, but it's really just an overgrowth of a small group of bacteria. We have then used this program Decontam, which is originally developed for removing laboratory contaminants, but we've used a training data set of archaeological bone and commonly observed environmental taxa in archaeological skeletal samples, and we've used this to then further clean up the data set and remove some of these post-mortem blooms that were residual. After we have our cleaned up data set, you can see the results here, and if we plot this in principal coordinate space, you can see that our dental calculus plots with a dental plaque and away from gut microbes, gut microbial communities, and also environmental samples. And so this was a way of cleaning up this data set before proceeding. We also looked at authentication measures. And so we looked at to what degree we had damage patterns in our samples. And so you can see that in our ancient samples, we have very nice damage patterns across a range of taxa, whereas at our modern day samples, for example, at the bottom here, we have no damage. And we've repeated this and also looked at many other metrics like edit distance and sequence identity as we go through the next several steps. All right. So next I want to talk about composition. So now that we've identified our well-preserved set, what can we analyze from it? So this is looking at the most abundant taxa within this data set. So we're looking, you have rows here are different bacterial species, and columns are individuals, and they're color coded. And what we can see is, this is our group, howler monkeys and gorillas clustered together. This is in a hierarchical clustering. They have similar oral profiles, oral communities. Then we have chimpanzees, and then we have humans and Neanderthals, which are quite mixed. And overall, what we see is that the primate oral microbiota cluster by host genus and other factors, but don't strictly recapitulate phylogeny. We can then look within Homo, for example, to see if it's been previously proposed that Neanderthals have a distinct structure. We didn't find that. We found that members of Homo actually have highly similar oral ecologies. And we can look at this in a number of different ways. But generally, when we're looking at the community structure, the ecology, Neanderthals and modern humans are not substantially distinct. We can then look within modern humans. And we've done this here, and to look to see to what degree we might have structure within locations, or geographic locations. So here we have samples both from Africa and from Europe, and also from different time periods. However, what we find is that the human oral microbial ecology is not substantially structured by time, geography, or lifestyle. This is really interesting because this is very different from the gut microbiome. The gut microbiome is heavily influenced by these factors, and we see vast differences in the microbial ecology based on these factors. We do not find that the oral microbiome tracks these same metrics. This makes sense in many ways based on the lifestyle of the oral microbes, which differs substantially from the gut microbes. So for example, the primary nutrient source for the gut microbiome is diet. And so things like diet and lifestyle heavily influence its microbial composition and its ecology. Whereas in the oral microbiome, the primary nutrient source for most microbes is actually saliva and other mucins. And so it's a much more stable community. We next wanted to look at the core microbiome, to look at what bacteria, are there bacteria that are core, that are invariant within a species, or maybe shared by multiple host species. So we wanted to see to what degree we might have conserved taxa across evolutionary time. We use upset plots to take a look at this, and we found that there are 10 genera of bacteria that are core across anthropoids, so are shared. These are them, and I'm sure these core oral microbes have been conserved for over 30 million years to retain within the primate microbiome over this period. However, I'm sure from this list, you're not as intimately familiar with them as I am, and so it can be helpful to look at them in different ways. One of the things that's really important about the oral microbiome, and it's distinct from other sources, is that it's highly structured. It has spatial relationships that form in a very structured way. This is an example of an investigation done by Gary Borsey's lab on the very complex structure of the oral microbiome. These microbes have evolved with each other and actually have evolved receptors that bind onto each other to form a structured oral biofilm. And we were very curious to see to what degree these relationships have been conserved through time. So here we've constructed a model of the human oral microbiome, and we wanted to know, well, which microbes are those that have been conserved over 30 million years, and which ones are perhaps more recently have become core to the human oral microbiome. And so we've color-coded them here. And you can see actually that what I find really interesting is that the core primate oral microbiome contains major structural taxa. So the major taxa involved in initiating biofilm formation, bridging taxa that allow later colonization, and then the outermost layers, those are all part of the core anthropoid oral microbiome and have been with us for a long time. And it's more of these other members that play subsidiary roles that have more recently associated with us, or have more unstable or more variable associations. One thing that was really interesting to me, though, is that among these 10 genera that have co-evolved with us for at least 30 million years, they're vastly understudied. So the ones that are shared among anthropoids are Campylobacter, Capnocytophaga, Otowia, Pseudoprobionibacterium, Selenomonas. These 5 of the 10 have almost no study on them. We know very little about them. Similarly, for great apes and humans, the Aneryllinaceae is a family. It doesn't even have named genera or species. It's so poorly understood. Fredibacterium only has a single genome. Olsonella, barely understood, and even Tannerella, probably the best studied of all, still is vastly understudied and only has a few published genomes. And the same is true for chimps and humans. TM7 is an entire phylum that has very few named species, and we know very little about the other bacteria as well. So although they're known to be oral associated, they're clearly important and they've co-evolved with us for so long. They're dramatically understudied by oral microbiologists who have instead focused a lot of their efforts on what they consider to be clinically relevant taxa for caries and periopathogens. However, it may be that studying these taxa that are key to the formation of the biofilm will lead to new insights in trying to understand how to maintain a healthy oral microbiome structure. One thing that's also interesting is that we see there are 3 clades of periopathogens. These are groups of bacteria involved in the advancement of periodontal disease. These are not specific to humans. We actually find that they're shared amongst other primates, either amongst all of the great apes or shared with chimps. And next, we're going to look at phylogenetics. So one of the things we wanted to know is to what degree the microbes within a host species are going to track the phylogeny of that host species. And so what we found overall is that we do have relatively good clustering by host genus. But one thing that we found was quite interesting is that Neanderthals do tend to cluster together. And in this case, we had one Paleolithic, upper Paleolithic individual in this analysis from El Morone. In every case, the El Morone individual also clustered with the Neanderthals. We then expanded to look at, there's 8 core taxa that we were able to perform this analysis on that had sufficient number of reads across enough individuals to do this. And in all 8 cases, El Morone always clusters with Neanderthals, which we found to be really interesting. We then expanded to look at a broader data set where we actually don't have quite as much data. So it's a data set where we have fewer reads, or we don't have as deep of sequencing, but we have more individuals. And we saw a similar pattern in that El Morone continues to cluster with Neanderthals, and then also other upper Paleolithic humans tend to as well, from Rigney, as for example. And so we see this consistent shared genetic diversity in core taxa among Neanderthals and upper Paleolithic humans that might be related to the sharing and the persistence of Neanderthal strains into human populations long after Neanderthal extinction. And last, I want to talk about functional aspects of the oral microbiome. So looking across primates, for example, we looked at Tannerella forsythia. This is a periopathogen involved in the advancement of periodontal disease. And we looked at the different virulence factors, which are shown here in the rows. And the individuals are shown color-coded, in the rows, and the virulence factors are shown as columns. What we can see for Tannerella forsythia is, in general, the virulence factors are present for all host species. So for all host taxa, they have Tannerella forsythia in this case, and they also have the whole complement of virulence factors. Another periopathogen, Porphyromonas gingivalis. This is a species that has many different strains of very different virulence. Here we saw a different pattern where we saw that in some individuals, we see the either absence or great reduction of genes that are involved in a specific type of virulence. So in this case, for example, the MFA1 genes, which encode fimbriae, which are important for cell attachment and are an important part of the more virulent strains, are absent in the howler monkeys and 3 of the 4 chimps and 1 of the 2 humans we analyzed. We also see HAG-A, which is a hemagglutinin, is absent in the howler monkeys and in 1 of the modern humans. So what we see is that most periopathogen virulence genes are present among all anthropoids, but there is variation within taxa. This might actually help us understand strain variation and the evolution of different virulence strains through time. We then looked also at more broader categories of function, and we noticed that humans segregate on the basis of carbohydrate metabolism. We have we're distinct in the carbohydrate metabolism of our communities. And we also found that there are functional differences between human and non-human oral microbiomes that are primarily driven by genes in Streptococcus. This was initially surprising to us because Streptococcus was one of the taxa that was initially flagged as this highly conserved taxon. It's co-evolved with us for over 30 million years, and it's present throughout our samples. So we decided to look into this a little bit more closely. Within Streptococcus, there are subclades. These are not species, but these are clades of species that are functionally distinct within this very large genus. And so we decided to then look to see if there were differences in these clades amongst our host species. So when we plot that, what we see is actually we see really large differences in these clades of Streptococcus. So Streptococcus as a whole is not terribly different amongst these, but when you start to look at the subclades, they are. What's particularly interesting is that you'll notice that for humans, and most humans in particular, you see a great expansion of 3 subclades. So you see the sanguineous group, the mitis group, and the salivarius group are vastly expanded in most humans and Neanderthals. Oops, sorry. What's important to that is that these clades today in humans are known to have the capacity to bind salivary amylase and to use it for starch digestion. This is a quite unique feature of these bacteria. It's an ability that is not possessed by any of the oral taxa in the mouth. So it's quite specific. The ability to use salivary amylase for starch digestion is specific to these 3 groups. And this, for me, immediately raised the question that was brought up earlier, which was first proposed by Leslie Aiello, which is the expensive tissue hypothesis. The idea, as it was explained earlier, is that at a certain point in our evolution, our brains really start to grow and our guts begin to shrink. And the only way that you can really explain this is that we have a dietary shift that allows us to have a much more energy-rich diet with less effort for digestion. And so there are 2 main theories proposed for what could explain this. The first one, the earlier one, is really this raw meat hypothesis. And Catherine Milton has gone to great lengths to kind of articulate this hypothesis. And the idea being that hunting and meat consumption was an early driver of the growth of brain size in Homo. An alternative hypothesis is the cookivore hypothesis proposed by Richard Wrangham, which is that actually it's the ability to cook, and specifically to be able to cook carbohydrate-rich foods, that is the driver of large brains. And the key to this hypothesis then is the early utilization of underground storage organs like bulbs, storage roots, and tubers. We see today that many hunter-gatherer groups around the world rely on USOs as a major nutrient source. And there's a number of primates that also rely on these. So for example, in baboons are highly reliant on underground storage organs. And we also know that salivary amylase has undergone copy number expansion in humans. So chimpanzees only possess 1 copy of salivary amylase gene. To the best of our knowledge, Neanderthals and Denisovans only have 1 as well, although there aren't that many that have been studied in that way. But in humans, we see a big expansion in salivary amylase genes, which are thought to aid in our digestion of starches and a correlate to increased carbohydrate consumption. In addition, some of the work by Amanda Henry looked at the presence of starches within Neanderthal dental calculus, arguing that early starch reliance could have been important in Homo. And so we took a look at this and we wanted to understand, okay, we see that we have an expansion of these taxa that today have the capacity to bind salivary amylase and use it for starch digestion. But what's the mechanism? The mechanism is that these taxa, there's 2 genes, amylase binding protein A and amylase binding protein B, that are found within these within strains of these groups. And what they're able to do is effectively capture salivary amylase from saliva, physically dock it to their cells and use it to process starches to produce glucose for their own consumption. But it also plays another role, and that is because salivary amylase constantly bathes the teeth and is an important part of the acquired enamel pellicle, this proteinaceous cover that forms all of the enamel and is what bacteria use to bind on and initiate biofilms. So bacteria lack the capacity to bind directly onto enamel. Instead, they bind onto this protein layer that forms over the teeth. And in humans, because of our high alpha amylase expression, we have a lot of amylase that's actually coating and bound to the enamel. It spontaneously binds, and this allows then the bacteria that have this receptor to bind onto this amylase and initiate growth. And so we wanted to know to what degree we had evidence of amylase binding protein A or B within our data set. And what we found was, I thought, pretty exciting, is that we found strong evidence for amylase binding protein A in all of our modern humans and some evidence in Neanderthals. And we found good evidence of amylase binding protein B in both Neanderthals and humans. However, we did not find it at any substantial level, really at trivial levels that could be noise in the data set amongst the non-human primates. And so we see a sharp differentiation between Homo and other non-human primates with respect to the ability to capture salivary amylase by these streptococci. So Homo have the capacity to bind and use host salivary amylase for niche construction, for enabling them to form a biofilm and increase in number, and also for dietary starch utilization. And so I'm excited because I think that by studying the evolution of the oral microbiome, it's starting to give us insights, not only in these microbes, but in our own evolution and our relationship to our closest relatives. And so with that, I'll close with just saying that I'm extremely excited about the future of microbiome research in this field. As technologies advance, more and more is becoming possible. And I'm really hopeful that we will be able to investigate new and diverse questions about human evolution by studying our microbiome. And I want to thank all the individuals who are co-authors on this study. It took a real effort by many, many people to put together a data set like this and also to analyze it effectively. And so I want to thank everyone who is part of this study. Thank you. And thank you for your attention.